Hi, Michelle Glass here. Welcome back to another one of our Chapter 12, Lecture 12 videos here. Our topic is membrane potential. Membrane potential involves three different types of potentials. We have what's called the resting potential, the graded potential, and then the action potential. We've real briefly talked about the action potential as part of our skeletal muscle discussion. So you kind of know that that's the electrical signaling that's happening with neurons, and we'll have an entire presentation just on the action potential. So our job in this video is really to talk about the other two. Your resting potential is based on the fact that your intracellular fluid, your ICF, and your extracellular fluid, your ECF, are different from each other. And they're different because the membrane of the cell is selectively permeable. So we know already about the sodium-potassium exchange pump. We know that this is part of the homeostatic mechanism maintaining the normal balance in the body. The cells are pumping all the time, 3 sodium ions out and 2 potassium ions in. So this is setting up a huge concentration gradient where you have a ton of sodium in the ECF, and then you have potassium inside of the cell. So because you have this difference in how much positive charge you have across the membrane, we're going to be seeing anything with a negative charge hanging out right inside the membrane because it's super attracted to the positively charged sodium that's in the ECF, because remember, opposite charges attract. So that when we actually measure across the membrane, our resting cells have a slight negative charge. Now this negative charge varies cell type to cell type, and our textbook tells us that the neuron's resting membrane potential is negative 70 millivolts. So that's the number that we're going to be learning in this class. Now, ion channels can actually change the resting potential because it's allowing for the movement of ions. Leak channels are always open. Chemically gated channels are opened or closed, or more accurately, let's say activated and deactivated by the binding of a chemical. And then voltage gated channels, we already know, can be activated or deactivated, opened and closed, based on changes in the charge or the voltage across the membrane. Your voltage-gated channels are going to be special to excitable membranes. So we saw these on the sarcolemma, the cell membrane of our skeletal muscle, and you would see that too on your axolemma. That's going to be the cell membrane of your axon. And we have some other excitable membranes in the body as well, cardiac muscle, smooth muscle, and so forth. Now, graded potentials can't travel very far from the stimulation site, and they're happening based on activation and deactivation of chemically gated channels, so that's important to note. Graded potentials can be really big or really tiny, so there's some variation here in your graded potentials. In the graded potential, we're looking here at our axon hillock. So we're looking at the area where the axon comes off of our cell body of our neuron. And we already mentioned that in the axon hillock, we have chemically gated sodium channels. So this is very similar to what you saw in skeletal muscle contraction at the neuromuscular junction. You had the motor end plate that contained chemically gated sodium channels. We're going to see a synaptic knob will release acetylcholine. That's our chemical that binds to our chemically gated sodium channels. Those channels open and we get an influx of sodium into our cell. And the reason is because we have set up this high concentration of sodium outside of the cell. You open up these channels and sodium floods in. If you get a big rush of positively charged sodium into your cell, you're going to see that our cell depolarizes. It becomes more positive. It's becoming more like the extracellular fluid. There's less of a difference here. There's less of a polarity. So this is your graded potential. this initial influx of sodium through chemically gated sodium channels that are being opened up based on the binding of acetylcholine or some other neurotransmitter. What we see happening next is that that sodium is going to move through the cell, it's going to diffuse, and we call that diffusion of sodium inside of the cell local current. So it's not hanging out right there, right at the axon hillock where it comes in, but it is going to spread a little bit away from that. And that will come up later in our conversations. Your graded potentials can either depolarize your cell, which is what I've drawn for you already, meaning to make the cell more positive. This is happening with an influx movement in of sodium ion, or you can hyperpolarize the cell, which means to make it more negative than resting. This can happen by an efflux, an exit of potassium ions. So we can see either one of these things happening. Graded potentials lead to action potentials, which is the topic for our next video. The action potential is a cycle of depolarization, becoming more positive, repolarization, moving back to resting, and then hyperpolarizing, becoming even more negative than resting. So stay tuned for more.